Good morning, class. Today we are going to learn the last part of chapter number 6, life processes. Let us see what is meant by blood pressure. Blood pressure, as the name indicates, it is a force that blood exerts against the walls of a blood vessel, where blood usually flows within a blood vessel. So, while flowing, this blood, it exerts its pressure on the surface of blood vessel. This force is nothing but blood pressure. This pressure is more or greater in arteries compared to veins. Can you tell me why? Why? Because the force of blood is very high in arteries compared to veins. Why? Because the veins always carries blood against the gravity from the different parts of body towards the heart. Whereas in case of arteries always carries blood from heart to different parts of the body towards the gravity. So the force is always high in arteries compared to veins. What is meant by systolic pressure and diastolic pressure? As you know, what is meant by systole? Systole, contraction of heart or cardiac muscles or heart chambers. Diastole, relaxation of heart muscles or heart chambers. What is meant by systolic pressure? As the name indicates, the pressure of blood inside the artery during ventricular systole or contraction. So, systolic pressure is always measured only the contraction of ventricles, the lower chambers of a heart. So, the pressure of a blood which is created after the systole of or during the systole of or contraction of ventricles. Next, diastolic pressure. The pressure of blood inside the artery, only in arteries, during ventricular diastole. When ventricles relax, the pressure which exerts the pressure the blood which exerts the pressure at this time during ventricular diastole, that pressure is nothing but diastolic pressure. So, what is the normal systolic pressure? What is the rate? Let us see now. The normal systolic pressure is 120 mm of Hg and diastolic pressure is 80 mm of Hg. You know the instrument which is used to check blood pressure or test blood pressure? Yes, anyone? It's sphygmomanometer. Sphygmomanometer. You might have seen this. Um, they used to there will be a one pump which is used to press, and within a sphygmomanometer there will be a mercury which will rise its number. It shows its number. Actually, this is an older instrument. Nowadays we can see electronic instruments. So I'm telling about sphygmomanometer, the older instrument. Next, what is the normal systolic pressure? It's 120 mm of Hg. Hg here. Why the name Hg? What is meant by Hg? Mercury level. Mercury will show 120 millimeter. And diastolic pressure is 80 millimeter of Hg. If the reading is 120 mm Hg out of 80, then your blood pressure is normal. If it is less than 120 or 80, then it is lower blood pressure. If it is above 120 and 80, then it is higher blood pressure. Now let us see what is meant by excretion. It is a biological process of removal of harmful nitrogenous metabolic waste from the body is called excretion. Excretion or egestion, both are same. Removing out of waste products. The waste products are also called nitrogenous substances. Why the nitrogenous the word? Because the waste substances always has nitrogen compounds. So especially the proteins, unwanted amino acids. Next, in unicellular organisms, excretion takes place by the process of diffusion, and even in few multicellular organisms. And multicellular organisms use specialized organs to perform same function. What are those specialized organs? How we excrete? Say for example, urine. We excrete urine through, what are the filtration units of urine? Kidneys. So excretory unit is kidney. And anus, nitrogenous waste excreted out, solid waste is excreted out through anus. So these are specialized excretory organs. And carbon dioxide, which is an excretory product, excretory gas, exerted out through nose via the lungs. Next, excretion in human beings. Let us see excretion in human beings. We have a pair of kidneys, a pair of ureters, and a urinary bladder and a urethra constitute human excretory system. Let us see the picture. This is a picture of human excretory system in which we have two kidneys. Kidneys are present on either side of the backbone. Don't show your frontal portion. Actually, the kidneys are present on either side of a backbone, back. In the abdomen, especially in the abdominal region. Kidneys produce, what is the main function of kidney? It produces urine. How? By filtering, yes, blood. The main function of kidney is it filters the blood, thereby it produces the waste products, urine. Kidneys produce urine and urine from the kidneys passes through the ureters. Can you see the picture? Yeah, two kidneys. Next to the kidneys, the continuation part is tube-like structures called ureter. Both the ureters, or either side of the ureters, enters into urinary bladder. Urinary bladder is a muscular organ where it stores temporarily urine. When after the formation of urine in the kidney, the urine enters into ureters. From the ureters, urine enters into the urinary bladder where it temporarily it stores and excreted out when it is filled. So let us see the last point again. Kidneys produce what? Urine. Urine from the kidneys passes through the ureters into the urinary bladder and remains stored there until it is released through the urethra. Urethra is a small opening present at the end of urinary bladder. It will relax or open when urinary bladder is filled. This is the structure of nephron. What is meant by nephron? It is a structural and functional unit of kidney. Nephron is a cell. A million number of cells are present within a kidney. Those cells are nothing but nephrons. This is a single nephron, a single cell. See the structure. See the red color pipes. The red color indicates the red color blood vessels indicates arteries. The blue color one indicates veins. Always red indicates arteries, blue indicates veins. So what is the main function of nephron? As I told you already, it filters the blood. If the blood has more concentration of amino acids, which is not required for our body, more concentration of water, excess of vitamins, excess of minerals, all these things should be eliminated out from our body. So this elimination or filtration, the blood filtration or purification takes place within nephron. Where exactly? See, the arteries first brings blood towards the nephron. And here arteries, it form a capillaries. The branches of the arteries is nothing, they are nothing but capillaries. A group of capillaries called glomerulus. You can see the structure of glomerulus. The glomerulus is present within a C-shaped or cup-shaped Bowman's capsule. This Bowman's capsule is, there is a extended part in a Bowman's capsule. This part is called tubular part. So the end stage of a tubular part, there is a collecting duct. See the structure. First, blood vessel or artery. The branches of artery is nothing but capillaries. A group of capillaries, a cluster of capillaries forms a glomerulus. Glomerulus is present within a C-shaped structure called Bowman's capsule. The next part of a Bowman's capsule is tubular part. The end part of this tubular part is collecting duct. Tubular part is divided into first PCT, proximal convoluted tubule, next Henle's loop or loop of Henle, next DCT, distal convoluted tubule, finally collecting duct. Structure is not there in your syllabus, only the function. What is nephron? What is its function? Where filtration of blood takes place. Where filtration happens? Within a glomerulus. Glomerulus is present within C-shaped structure called Bowman's capsule. Let us see now. Urine is produced to filter out waste products from the blood. Nephrons are basic structural and functional unit of kidney. The cluster of thin-walled blood capillaries called glomerulus in the nephron. It filtrates the urine, and each cluster is associated with cup-shaped end of the tube called Bowman's capsule. See, here blood filtration takes place within glomerulus. It is present within Bowman's capsule. After that, the waste substance or the filtrate enters into the tubular part. From the tubular part, finally the waste product or the waste fluid or urine enters into collecting duct. From the collecting duct, it enters into the middle portion of a kidney called medullary region. From there it enters into ureters. From ureters to urinary bladder and then urethra. Substances like, what type of substances are excreted out? Let us see now. Substances like glucose, amino acids, salts and a major amount of water is selectively reabsorbed from the initial filtrate as the urine flows along the tube. Once filtration happens, after filtration, if the water or any salts are excreting in more concentration, then what will happen? The tubules will reabsorb it back. Reabsorption depends on the amount of excess of water in the body and dissolved waste to be excreted. The urine from the kidney enters the long tube called ureter. Ureter connects the kidney with the urinary bladder, and the urine from the kidney is passed to urinary bladder and stored in the bladder. This I have explained already. When the pressure in the expanded bladder leads to the urge to urinate, the urine is passed out through urethra. This signal is given by CNS, central nervous system. This you will study in the next chapter. Next, excretion in plants. Just like in humans, even plants excrete. But there is no specialized excretory system. Let us see what is that now. Excretion in plants. Oxygen, it is a waste product of photosynthesis, is released out. Oxygen is very important for humans for respiration, even for plants. But oxygen as an end product during photosynthesis. During photosynthesis, oxygen is released out. It is an excretory product. It should be released out. So, excess, you know how oxygen is released out? Through the stomata and lenticels. Excess of water should be removed out. It will take place through transpiration. Some waste products may get stored in the leaves which will fall off. Few waste products or few waste substances, it gets stored in the leaf and after that the edge will become yellow and it will fall off. Many waste products are stored in vacuoles. As you know, vacuoles' main function is storing excess of food materials as well as waste products. Some waste products are stored as resins and gums in old xylem. You might have seen some kind of gummy substance which is present on the surface of bark of a tree. So those are called resins and gums. Plant excrete some waste materials into the soil around them through the roots. So this is about excretion in plants. Now let us see what is meant by hemodialysis. As the name indicates, hemo means blood, dialysis, filtration. What is meant by dialysis? Why we should follow this technique? Let us discuss now. When the kidney fails, we have two kidneys. If any one is working properly, it is more than enough to filter the blood and to form a urine. But when both the kidneys are not working, what will happen? The nitrogenous waste or as well as toxic substances concentration will be more in the blood and there is no net formation of urine. So the blood is impure now. It should be filtered. How this will happen? Artificially we can do this function through the process of hemodialysis. What is meant by hemodialysis? When the kidneys fail, it results in a lot of complications, and to compensate this situation, a technology called dialysis has been developed. It uses a machine filter called dialyzer or artificial kidney. As it performs the function of the kidney, it is also called artificial kidney. This is to remove excess of water and salt. What will happen during dialysis? It removes excess of water and salt and it also balances the electrolytes in the body, and it also removes waste products of metabolism like excess of sugar, excess of amino acids, excess of fats, like that. Blood from the body is removed and flowed through a series of tubes made up of a semi-permeable membrane. Within dialyzer or within dialyzing unit, you can see a series of tubes which just looks like a blood vessel, and it is made up of a semi-permeable membrane. You know why the semi-permeable membrane we are using word here? Because it's selectively permeable. It allows only few substances but not all. So through the process of semi-permeability, it allows only impure substances. By maintaining balance in the body, it filters the blood within a dialyzing unit. How blood is removed from the body? Through the vein. So there are two openings. From one opening, blood is drained out. Through the other opening, the purified blood is allowed to enter into the body of a patient. A vein. Through the vein, we are draining out the blood, and after purification within dialyzing unit, we are allowing purified blood through the artery into the body of a patient. A dialysate flows on the other side of the membrane, which draws impurities through the membrane. So the membrane main function is which draws or which drains impurities from the blood. This is about hemodialysis. Hemodialysis is one of the artificial technique which is performed in the patients in which both the kidneys are failed. What will happen during hemodialysis? Blood is drained out from the patient, and it should be cleaned within a dialyzing unit, and after that the purified blood is entered into we are allowing that purified blood into the patient's body. This is about hemodialysis. Next, let us see the activities. This is activity number 1, 6.1. In your textbook, the activity number is 6.1. That is why I am giving the same number. What is this activity? To show that chlorophyll is essential for photosynthesis. I told you already, during photosynthesis, chlorophyll is very, very important. Why? Because it traps sunlight. Without sunlight, there is no photosynthesis. Let us see how we are going to conduct this experiment or activity. Materials required. What are the materials? A potted plant of variegated leaf. Variegated leaf is nothing but which have a different colors. A leaf with variety of colors, not only green, with different colors. White paper sheet, pencil, beaker, water bath, iodine solution, alcohol. These are the materials which are required for this activity. First step, what we have to do? A potted plant with variegated leaves is placed in sunlight for about 6 hours. Take a potted plant with variegated leaf and it is placed in a sunlight for how many hours? 6 hours. Next, pluck a variegated leaf from a plant and trace the outline of this leaf on a piece of paper. Next, dip the leaf in boiling water for few minutes and then immerse in the beaker containing alcohol and boil it in a water bath till it decolorizes. Water bath is nothing but boiling water. A vessel with boiling water. You can consider it as a water bath. The leaf is then dipped in a dilute solution of iodine for a few minutes. Take out a leaf now and rinse off the iodine solution. Observe the changes in a color. What are the observations? Let us see the observation now. The leaf has two types of patches, bluish black and yellow. The bluish black area contains starch and the yellow areas without starch. If you see yellow area, it indicates there is no starch. If it shows blue color or blue area, it indicates the presence of starch. Bluish black areas are the ones which were green previously, while non-green areas remain pale colored. Conclusion. What is the conclusion of this experiment? It proves that only chlorophyll containing areas, that is green parts of a leaf, produce starch, which is a product of photosynthesis. Thus chlorophyll is essential for photosynthesis. You can check this. Take a green leaf and non-green leaf. So if you conduct experiment for green leaf, you can see blue black color. See the second picture, blue black color. Blue black color indicates the presence of starch. So Due to the presence of chlorophyll pigment, leaf appears green. As the leaf is green, as the chlorophyll pigment is present, it conducts photosynthesis by entrapping sunlight and gives blue-black color. It shows the presence of starch. What about the non-green color leaf? As the chlorophyll is absent, there is no photosynthesis and it won't show any blue-black color, and it indicates the absence of chlorophyll and the absence of photosynthesis. Now, next activity 6.2, to show that carbon dioxide is necessary for photosynthesis. Along with chlorophyll pigment, carbon dioxide is necessary for photosynthesis. Carbon dioxide reacts with water to give or to produce glucose. Let us see the materials which are required for it. 2 healthy potted plants of same size, 2 glass plates, 2 bell jars, vaseline, watch glass, potassium hydroxide, KOH, alcohol, spirit lamp and beaker. What is the procedure? Keep 2 potted plants of the same size in a dark for 3 days so that leaves become free from starch. Why we are keeping both the plants in a dark? Because the reserved food material starch should be utilized by the plant, so that we can conclude there is no starch now in these plants. Why? Because it is already used up by the plants. Place the potted plant A on the glass plate and put a watch glass containing potassium hydroxide. In the first setup A, we can see the plate along with KOH. The KOH, potassium hydroxide, main function is it absorbs carbon dioxide by the side of a pot and cover it with a bell jar. Place the other plotted plant on a second glass plate and cover it with a bell jar. See, you cannot see any type of KOH in second plant or second bell jar. Vaseline is used to seal the bottom jars to the glass plate so that the setup is airtight. Both the plants are kept in sunlight for 2 hours. Now, pluck a leaf from each plant and test the same for the presence of starch. The first setup A, it won't show any starch, but in case of B, you can see starch. Why? Because in setup A, the potassium hydroxide absorbs carbon dioxide, so there is no availability of carbon dioxide for photosynthesis. But in case of setup B, there is no absorption of carbon dioxide as there is no KOH, and it shows the presence of starch. Why? Because due to the availability of carbon dioxide. What is the observation? The leaf of plant B without potassium hydroxide turns blue-black, while the leaf of plant with potassium hydroxide remains pale colored or colorless. What is the conclusion? This experiment demonstrates that leaf plant B has synthesized starch with the help of photosynthesis, and leaf of plant A has not synthesized starch as it does not contain carbon dioxide, as the same is absorbed by potassium hydroxide. So, photosynthesis did not occur in A. Now, third activity, 6.3, action of saliva on starch. As I told you already, saliva has salivary enzyme, or ptyalin enzyme. It acts on starch or any complex sugar and converts them into simple sugar. Let us see now. What are the requirements? What is the procedure? Take 1 ml of starch solution, 1% starch solution in 2 test tubes, both test tube A and B. See the picture, A and B. We have taken 1 ml of starch solution. Add 1 ml of saliva to the test tube A. Add 1 ml of saliva to the test tube A and leave both the test tube undisturbed for 20 to 30 minutes. We have not added anything to the test tube B except starch solution. Now, add few drops of dilute iodine solution to the test tubes. Both the test tubes add iodine solution. If starch is there, iodine converts the starch and gives blue-black color. If starch is not there, it won't give any color. That is, it won't give any blue-black color. Let us see now. What is the first question? In which test tube do you observe color change? Definitely, you can say we will observe a color change in test tube B. Why? Because there is only a starch. Here the starch source is flour. You can take any flour. Say for example maida. So as the flour or starch is undisturbed and there is no enzyme in it, So there is no change in the starch. That is why when it reacts with iodine, it gives blue color. Whereas in test tube A, saliva has salivary enzyme. Salivary enzyme acts on this starch and converts them into sugar, maltose, or simple sugars or disaccharide. That is why when this simple sugar reacts with iodine solution, it didn't give any color change. Whereas a test tube A will show no color change as due to the addition of saliva, starch is converted into sugar. Question number 2. What does this indicate? What about the presence or absence of starch in the two test tubes? It indicates that test tube A does not contain starch, whereas test tube B does contain starch. What does this tell us about the action of saliva on starch? It tells us that an addition of saliva in starch solution, the starch gets converted into some other product. Here, maltose sugar. Maltose is a disaccharide, 2 sugar units. But glucose and fructose are monosaccharides, simple sugar or single moiety of sugar. Last activity, to show that carbon dioxide is produced during respiration. Materials require 2 test tubes, freshly prepared lime water, syringe or pitchcarry. Pitchcarry and syringe both are same. Rubber tube and a glass tube. What is the procedure? Pour freshly prepared lime water in each test tube A and B. Blow air in test tube B through the lime water with the help of a glass tube. Here one boy is blowing air. In your air, from that excreted air or blowing air, you can see carbon dioxide. Pass air through lime water in the test tube A with the help of syringe or pitchcarry fitted with a fine rubber tube. Here through the syringe we are passing a normal air or a fresh air. Now let us see what is the observation. Lime water turns milky in B, showing that we exhale carbon dioxide. In test tube A, lime water take long time to turn milky. Why? Because fresh air may have carbon dioxide, but its concentration is less compared to exhaled air. Conclusion. Lime water turns milky when carbon dioxide mixes with water. Lime water, when combines with carbon dioxide, it turns milky. Air blown out from the mouth contains carbon dioxide. The concentration of carbon dioxide of exhaled air is more compared to the fresh air. That's why the test tube B gives immediate milky result, but in case of A it gives it will take time to give milky. Why? Because the carbon dioxide concentration is very less compared to B.